Hi. Today I'll be talking about macroscale brain activity as waves shaped by cortical geometry as part of this symposium. Basically, this talk will revolve around this recent publication that we have, and you can scan the QR code for further details, but obviously I can't talk everything about the paper here, so you can attend the following sessions later today if you want to learn more about the paper itself. So basically, the paper focuses on trying to understand how the anatomy of the brain can constrain its emerging function. And what do I mean by anatomy? We can view the anatomy of the brain in two different ways. One would be the discrete view of the brain, where you partition the brain into isolated regions, and you try and estimate the connections between these regions and assume that the activity emerges from how signals propagate between nodes in this network. And this has resulted in quite, advancements in the last couple of years, where people have found that if you compare structural connectivity and functional connectivity, you will have this, you will find this strong coupling between the two. The limitations of the discrete view of the brain is that if you look at the neural tissue at macroscopic levels from millimeter to centimeter scales, it is actually not discrete. It's pretty continuous. And if you look at fMRI activity as well in those scales, you will see these waves of activity that propagate throughout the brain. And additionally, the discrete view ignores the idea that the brain is an intrinsic, real, physical object. And so how do we capture this continuous view of the brain? A lot of empirical studies have shown that even if you just observe, say, optical imaging in mouse, you can actually see these continuous patterns of activity that emerges in just resting state, or even in task fMRI in humans. And a class of models called near field theory has enabled an approximation of these meso to macroscopic scale dynamics. Basically, near field theory investigates the activity of populations of neurons as a whole. Again, not the activity of single neurons, but populations of neurons, and observe the emerging activity from there via the assumption that these populations of neurons are connected via a really simple connectivity kernel, W, which is mainly local connectivity, basically a local neighborhood type of connectivity, plus an exponentially decaying rule connectivity, where points really, really far from you would be weakly connected, as opposed to points that are very, very close to you. And this agrees with empirical observations, even in human diffusion MRI, where if you plot the weight of connections between regions as a function of their fiber length connectivity, you would see this exponentially decaying kernel. More importantly, near field theory approximates the activity of, approximates macroscale activity as waves that propagate following this damped wave equation, wherein you have the firing rate of a certain population affecting the emerging activity of another population via a damping like activity. Similar to, say, when you throw a pebble into a pond and you see the ripples that propagate but decay in space and time. And the important aspect of this damped wave equation is this spatial kernel here, which is the Laplacian, which dictates, which suggests that the emergence of activity heavily relies on the physical geometry of the substrate, basically here the cortex, and its local connectivity. Now we have these two viewpoints of the anatomy of the brain, one from a discrete point of view, and one from a continuous point of view using the actual geometry of the brain. Now, how do we capture the structural constraint? Now, in physics and engineering, this is a common problem where you can investigate these constraints by what you call as the eigenmodes. basically every system would have its own eigenmodes, which correspond to its natural spatial patterns of vibration or excitation. So take, for example, a violin string, and if you pluck it, you would see the emergence of these distinct patterns that vary if you change the frequency of plucking of that particular string. Now, these spatial patterns are pretty dominant, are pretty fundamental for the system, and you can capture the family of these eigenmodes, and it can fully describe any emerging activity of that particular system. Another important note to say is that these eigenmodes are very, very sensitive to the system's physical properties. So for example, a string again, the frequency of the first mode here on top heavily relies on, say, the tension of the string. That's why when you play a guitar, you can change the, where you put your finger to like the stoppage, and also in the density of the particular string, or even at the actual length. So this means that the physical property of a particular system heavily dictates the eigenmodes that emerge. So our paper investigates precisely this, that if you have two different properties, anatomical properties of the brain, you can verify and investigate the constraints provided by those anatomical structures. So in this case, again, we estimated the cortical surface of the brain via this triangular mesh obtained from free surfer. And we also obtained a high resolution connect time estimated from diffusion MRI. And to calculate the eigenmodes, we solved this simple Laplacian eigenvalue problem, where on the left, this is what we call the Laplace-Beltrami operator, which encodes the spatial relationships of the points or the nodes in the connector, for example. And when you solve this, kind of like an eigen decomposition, you will get a family of eigenvalues and a family of eigenmodes, and they are kind of a one-to-one correspondence. One eigenmode has its own intrinsic eigenvalue. And you can approximate the Laplace-Beltrami operator depending on if you start studying the cortical mesh or the connectivity matrix. By doing so, you can get these geometric and connector eigenmodes. And the beauty of this is they are actually arranged in a really peculiar manner, where the first mode would correspond to the lowest spatial frequency or the longest spatial wavelength. So you can see here that it covers the entire brain. And as you move further along, higher modes will now correspond to higher spatial frequencies or very, very short spatial wavelengths. So you can see here the patterns are very localized. Now, these eigenmodes are what we call as basis sets. Basically, they are independent, and you cannot capture one eigenmode by combination of two or more other eigenmodes. That means we can represent any empirical map, say Y, as a linear superposition of these eigenmodes weighted by some coefficient A. So basically, as an example here, for this empirical spatial map Y, if we find that the A coefficient for mode 2 is higher than the others, that it suggests that that mode 2 is more expressed in that spatial map than the others. And we did that mode decomposition and reconstruction using the Human Connectome Project fMRI data, both resting state and task-evoked. And here on the left, which I'm showing sample task-evoked spatial maps for the different tasks in HCP. And on the right, I'm showing how well we can reconstruct these empirical data using the geometric eigenmodes. And you can see that approximately 100 geometric eigenmodes, we can already reconstruct the data quite accurately. And if you span the number of eigenmodes, and you can see here that at 100 modes, we're already capturing a reconstruction accuracy of about 0.8 about 8%. Now, if you do that for both the geometric and connective eigenmodes, what we found is that the reconstruction accuracy of the geometric eigenmodes seemed to be better than the reconstruction of the connective eigenmodes. And we moved further and asked, how about brain dynamics? So what we did was to adopt the simple wave model of neural field theory and compared that to a highly sophisticated neural mass model that heavily relies on the connectivity matrix that derived from diffusion MRI. And we simulated resting state activity and stimulus evoked activity. And what we found that across different metrics that people use in model data comparison, we found that the wave model, even though it's really, really simple, performs just as much or even better than the highly sophisticated neural mass model. And when we do the stimulus evoked activity, we tried pinging, say, the visual cortex and allow it to propagate through time and space. We see this propagation. And if you look closer, it seems like with just a simple model, we can kind of capture this separation of the ventral dorsal streams, again, without, explicitly putting it in our experimental model. And if we follow the activity across the visual hierarchy, we find that there is this natural ordering of the responses that actually follows the hierarchy observed in the literature. And if we compare that to non-invasive estimates of this hierarchy, say via T1, T2, we find this pretty high correlation with the activity, the temporal ordering of the responses and the T1, T2 data. So in summary, what we found was, it seems like human cortical geometry can strongly constrain brain activity, and we only need about 100 out of potential 32,000 modes to get a pretty good accuracy. However, and the geometric modes seem to provide more parsimonious representation of activity than other anatomical modes, in particular the connective modes, because again, geometric eigenmodes are just simply derived from just T1 data without doing anything else. But however, I want to emphasize that this does not imply that complex connectivity beyond local and exponentially decaying rule connectivity is not important. The only thing that this says is that the contribution of this complex connectivity could be more subtle, right? We all know that diffusion MRI is highly noisy, so this could have affected our investigation, and probably the influence of complex connectivity would be more observable in resolutions not captured by fMRI, which is the focus of our study. And finally, we have shown that waves traveling through the cortical geometry can provide a really simple but highly effective mechanistic account for both data and physiological phenomena, even though we didn't actually bake it in the model. So finally, I just want to acknowledge the other authors of the paper, and it's a really multi-institute effort. And again, you can scan the QR code if you want to know more about the paper in detail. Thank you very much.